Hello, everyone, and welcome to the second ACS Medicinal Chemistry Letters Innovations webinar. I'm Lorraine Clark, a managing editor at ACS Publications, where I oversee 4 journals in the biological chemistry portfolio, including ACS Medicinal Chemistry Letters. We will begin the webinar today with a brief overview of the journal, the innovations article type, and introductions of the speaker, the topic, and the moderator. Okay. So moving on to the first slide. So ACS Medicinal Chemistry Letters was launched in 2010 with Dennis Leota from Emory University as the editor-in-chief. The journal publishes a variety of different manuscript types that are shown here, including innovations. I will talk about those more in the next slide, and welcomes manuscripts that pertain to a broad range of subject matter related to medicinal chemistry, such as compound design and optimization, biological evaluation, drug delivery, imaging agents, as well as pharmacology of both small and large bioactive molecules. Now, a quick background on the innovations manuscript type. So the innovations articles launched in 2010, and as you can see, they tell the backstory of drug discovery campaigns relating to the development of new therapeutic agents, and they also cover the backstory and development of new technologies that enable the identification of new therapeutic targets or advancement and acceleration of drug discovery programs. And so in this webinar series, we aim to highlight the research that is published in these innovations articles. So our moderator for today is Dani Schultz. She received her PhD from the University of Michigan in Professor John Wolf's laboratory, and then pursued a NIH postdoctoral fellowship at the University of Wisconsin-Madison with Professor Taeshig Yoon. Dani then joined Merck in 2014 as a member of Process Chemistry and Enabling Technologies in Rahway, New Jersey, and as of 2021, she's the director of the Discovery Process Chemistry Group in Kenilworth, New Jersey, where she's involved in the development of synthetic routes for drug candidates spanning HIV and oncology. For the webinar today, we would love to get you, the audience, involved in the discussion of today's webinar's topic. You can share your thoughts and questions in the Q&A panel, and we would address as many of them as we can after Eric's talk. And with that, I'm going to turn it over to Dani to introduce the topic and the speaker of today's webinar. Dani?
Thank you very much for that intro, and I'm really excited to be here today. And so welcome everyone. And so I'm going to give a little bit of background before jumping into Eric's talk today. And many of you on the audience are probably very familiar with how we go about discovering drugs. And it's through this design, make, test cycle. And maybe given that I am a chemist at heart, I naturally think that there has to be some sort of rate determining step along this. And what comes to mind is when we think about design, we have a lot of really great tools. We leverage computational chemistry, AI, maybe a little bit of intuition to kind of figure out what kind of molecules we want to make to ultimately test biology. And in fact, one of the first ACS webinars was with Greg Makara, who had a really great topic about using AI to help do scaffold hopping to get into the lead analog space. And what he really called out was that the attractive feature of this AI methodology was that it included synthetic feasibility. And naturally, what made me think is, okay, so for the make part, we're good here. We know how to make a variety of different molecules. But what many of us on the line probably know is that we tend to rely upon these battle-tested reactions because we know that they're going to work. And ultimately, what this can do is it can slow down this design, make, test cycle and ultimately limit the creativity of the molecules that we ultimately want to make. And so I'm going to be a bit provocative and say that maybe the rate determining step is going from make to test. And over the last 50 years, there's been a lot of really great methods in synthetic organic chemistry that have been developed. And as we look at where we're going, the use of biocatalysis, molecular editing, and HTE have definitely improved our ability to make different molecules. But when we look at the future, where are we? And I really loved this perspective by Kevin Campos, who is the VP of Process Research at Merck. He co-authored a 2019 science paper and basically goes into the fact that our understanding of biology is expanding, and as such, the chemical tools that we need to probe that are going to become a little bit more complicated, such as ADCs, PROTACs, peptides, etc. And ultimately, there's a growing recognition that we need innovations in synthetic organic chemistry in order to keep up with our pace and understanding of biology. And so it's almost a call to action that we need to invest in chemical synthesis. And how do we go about finding better synthetic chemistry methods? I would say that we need to roll up our sleeves and we need to start to engage, and we need all chemists on board to impact human health and really engage to begin to explore the chemistry communities to find the answers that we seek. And here's just a handful of a few different synthetic reactions that I think anyone would love to have a really robust route towards, such as sp3 coupling, CH activation, non-canonical nucleoside chemistry, bioorthogonal chemistry, and this is only just a few. I'm sure that there's much more. But ultimately, how do we find better ways to tap into this space? Well, if you look, there's a few different places to go. You can look external, and in fact, Reese and coworkers, along with a variety of other chemists and leaders in different pharma companies, co-authored this really great Nature Chemistry article in 2018, where they made the connection that there is a tremendous amount of really exciting synthetic organic chemistry occurring within academia. And ultimately, this could serve as a pipeline to feed into drug discovery to improve the types of chemicals, or to improve the types of molecules that we are ultimately able to access. But despite this being, a really great idea, there's still issues at hand. And this was actually the focus of a piece that Elsie Campo and I co-authored last year for Nature Chemistry. And it was the idea that adoption seems to be the issue. And a little bit of a shameless plug, if you're interested to hear more about this, this was episode 1 of our new podcast, if you wanted to hear more. And so specifically what I'm getting at here is that there is this notion that we have, this ivory tower where we have an academic saying, why don't you use my method? And then we have the industrial chemist at the bottom saying, well, because your scope has 100 examples and none of them are relevant to what I'm going to be working on. And so this adoption is definitely, I think, one of the main factors as to why we don't see, a better, connectivity to our academic partners. And so, there's this idea that, the industrial researcher is a little bit out of touch and doesn't really care about, the work of the academic chemist, and the academic chemist, has no interest in the adoptability. And obviously this isn't true, but it definitely, is something that we dove a little bit deeper into. And so the best way to overcome this is definitely to engage in these academic industrial partnerships. And I want to just kind of pull up a couple that I thought were really relevant because they get to some of these really meaty synthetic challenges that I highlighted up above. And so most recently there was a collaboration with the Sarpong group and Merck, where they found this really beautiful ring contraction chemistry to do this sp3 coupling. And then in 2020, the Barron Lab with BMS, developed this really robust route to making these non-canonical nucleosides. But if we look internal, there's a tremendous amount of knowledge inside our own walls and amongst the folks that we work with every single day. And that is the topic of today's talk, where Eric Voight at AbbVie is going to get into fueling the pipeline via innovations in organic synthesis. And what he brings about is this idea that we can build our own internal chemistry communities to tackle a lot of these synthetic challenges. Specifically, there is this gap that exists between discovery chemistry and process chemistry. And through the formation of these discovery synthesis groups, you kind of live at that interface where true innovation takes hold. And this is actually a group that I am part of as well at Merck. And what I would like to kind of end with before I kick it over to Eric is that, when you take these, internal groups and when you bridge them with an external partner, I think that you are truly unstoppable in the types of chemistry and molecules that you can access. And so with that, I'm going to turn it over to Eric, and I'll catch you all on the Q&A at the back end.
Thank you, Dani. So Dani did a great job introducing this topic, and I think for the next 40 minutes or so, I'd like to give you some highlights from an article we published earlier this year in ACS MedChem Letters. And the molecules I'll be talking about today are shown in this first slide. There's actually one unpublished story that I'll also be sharing today that's not represented here, or that is actually represented here, this chiral cyclopropane. So it's some unpublished work that I'll also discuss today. So before we jump into the chemistry, which is definitely going to be the focus of this talk, just wanted to go over briefly the agenda for the talk today. So I'd like to continue a little bit where Dani left off, just discussing the role of organic synthesis in the pharmaceutical industry. Also talk a little bit about the organization that I'm a part of, Centralized Medicinal Chemistry at AbbVie, and where my group fits into that organization. Then I'm going to go into 3 different case studies. The first will be by far the most detailed, with 3 different projects, including all contributing to cystic fibrosis therapeutic area. Then after the 3 case studies, I'll finish with some concluding remarks. So as Dani said, organic synthesis is often the rate-limiting step in the pharmaceutical innovation cycle. And there's 3 papers I think really well define this problem. So back 17 years ago now, Lipinski and Hopkins highlighted how vast the chemical space is, including just rule of 5 molecules, which of course doesn't even encompass all that we're interested in. Despite that fact, 12 years later, this mini perspective in JMedChem highlighted the fact that despite the vastness of chemical space, most medicinal chemists were still utilizing a very narrow set of reactions, the tried and true ones that Dani talked about. So over time, as we have insisted on the creation of more chemical matter that has high fraction sp3, larger molecules, as we're looking at new modalities to tackle targets that we had never considered before, all of these require more challenging chemistry. And Dani did a nice job highlighting this 2019 Science paper. The comment that I really liked in this paper was that continued innovation in chemistry is required to drive the discovery of the next wave of medicines. We certainly embrace that at AbbVie and believe that to be true. So looking at the continuum of a pipeline to discover drugs and bring them to patients, there are organic synthesis is an important part of this entire continuum. So here, I just have demonstrated on a timeline discovery, advanced preclinical, and then various stages of development. And along this continuum, you go from needing milligrams or even nanograms of material to really needing a long-term supply of the drug substance. And during this process of discovery and development, the requirements for the synthesis change very substantially. Early on, a diversity-oriented synthesis that can very rapidly prepare many analogs to answer key biological questions is the most important. And later on, really finding a low-cost and robust process for one specific drug molecule is the key. And along this continuum, the efficiency of the synthesis is increasing in importance, where the purity, the scalability, how long it takes to make all these molecules is really becoming more and more critical in accelerating this process. So historically, there have been really 2 buckets that organic chemists fit into. You have a sort of discovery medicinal chemists and process chemists. And while that really does effectively span this entire gap or this entire continuum, about 10 years ago, we recognized that there was a gap here where efficiency of synthesis is increasing in importance. And we thought, filling this gap with a group that's really focused on this space could be beneficial. So just looking at this, more detail. Really the old model where you have the start of a project, you have discovery chemists working hard to find the right drug, and then you have process chemists really trying to figure out how to make that one drug substance to bring a new drug to patients. This gap in the middle wasn't entirely clear. There was many different ideas of how this could be addressed. Outsourcing is one approach, although that can be sometimes slower than is suitable for the rapidly changing nature of projects at this stage. Certainly discovery chemists can fill this gap and scale up molecules even if that's not the primary role, or process chemists can move earlier and take on molecules that aren't nearly ready for development yet. But there was always kind of some question about who is best suited to fill this gap. So we started a group almost 10 years ago now that's really at the interface of discovery and process, and has helped to bring these together. And while we can by no means take full credit for accelerating the discovery of molecules that I'll present today, we think that having groups really focused on synthesis within discovery can help to accelerate the design synthesis data cycle. And hopefully I'll show you some examples of that today. So we have several discovery synthesis groups at AbbVie. One of them is within our centralized medicinal chemistry organization. So our strategy in medicinal chemistry, in CMEC for short, really tries to optimize all the components of this design synthesis data or design make test cycle that Dani introduced. So really the medicinal chemist is at the center of this cycle, trying to optimally specialize or bring in expertise from all of these different components and really also be a practitioner of all of these components of the cycle. So while historically there's been specialization, emphasis placed on the design and data parts of this wheel, there was, I would suggest, less emphasis on specialization and synthesis until more recent years. So over the last 10 years, I think the industry has been increasingly trying to emphasize more on specialization and synthesis. And our approach to that, as I mentioned, is discovery synthesis groups. So our group specifically focuses on rapidly finding and executing fit-for-purpose synthetic strategies for key compounds. So fit-for-purpose is key. We don't have to have the perfect synthesis all the time. And key compounds can mean an intermediate, final molecule, many different things, or even a diversity-oriented synthesis to many molecules. So in the context of finding synthetic strategies that are suitable for their purpose, we often will uncover new synthetic methods that really impact that project and also help to enhance our compound collection at AbbVie. A key part of this is collaboration and consultation with AbbVie chemists. We do a lot to bring all chemists together at AbbVie to try to address the most critical synthesis problems and to really grow and enhance our community. And also engagement with the external scientific community is very important, as Dani introduced also. And really the goal is to pursue global excellence in synthesis at AbbVie and across the industry. How we function day to day as embedded members of discovery teams. So this really allows us to focus on the science instead of resource allocation and prioritization. So everyone in the group, including myself, can spend the majority of our time in the lab solving scientific problems instead of worrying about whether we're making the right molecules or not. And we also really do what we can to smooth the transition to process chemistry. Since we're focused on how molecules are made, that helps in the transition from discovery to process. All right, so enough introductory material. I want to really focus the rest of the time on some case studies. And the first one will really illustrate what is our most common mode of engagement with discovery teams. So I'm going to spend the most time on that. So that is the cystic fibrosis therapeutic area. So cystic fibrosis, as many of you likely know, is a horrible, life-shortening genetic disease that's affecting an increasing number of people, many of who are in the U.S. So our ability to treat this disease has been improving, and life expectancy is now in like the 30 to 50 year range, which is a dramatic improvement over where we were not long ago. And that's very much due to the fact that there are highly effective treatments now, but still there's a great need for improved treatments. So cystic fibrosis is a multi-organ disease that is best known for its effect on the lungs, because that's often what is the cause of mortality. And it's caused by mutations in cystic fibrosis transmembrane conductance regulator protein, or CFTR for short. So to treat cystic fibrosis, a combination approach has been shown to be the most optimal, where a collection of potentiator and corrector molecules are combined to treat the disease. And the first approval that took this approach was Trikafta, which was approved a couple of years ago. And so we also have a portfolio of CFTR candidates. Among those is this potentiator 974, corrector 2222, which is now called galicaftor, and this C2 corrector molecule 3221. So our group was involved in all of these projects, and today I'll just talk about the C1 corrector and C2 corrector parts of this program. So C1 corrector 2222 is this chiral chroman molecule, and this was originally discovered through a screen of many different chroman amino amides, and of course these two aryl 4 amino compounds were initially a mixture of diastereomers and enantiomers that had to be separated tediously. And the team began utilizing an asymmetric conjugate addition approach to allow access to a large variety of enantiomerically enriched 2 aryl chromans, or flavanones, that could be converted onto the amines and then taken forward to give the final products. And this method really accelerated SAR exploration and allowed the team to find the best candidate as quickly as possible. So it's a really great medchem story that I encourage you to take a closer look at in this publication. So we needed an improved synthesis of this aryl cyclopropane carboxylate as part of this effort. So with that, we explored a new method that didn't exist at the time, a general method to couple a cyclopropyl anion, specifically this cyclopropyl Negishi reagent that can be obtained just from zinc insertion into the bromide. We found that this coupled with just incredibly well with a wide variety of aryl halides. So because this was typically done in 5 minutes or up to 3 hours at room temperature, we could tolerate a lot of Lewis basic or protic functionality that would be typically offensive to these types of reactions. And because it's so general, it's been applied across many different projects. Last we checked, was over 600 examples of being used at AbbVie, and we also know anecdotally that it's been used at other companies. We published this method a few years ago now. So to demonstrate its utility, we carried out single-step syntheses of 2 known molecules that had previously been prepared in 4 steps, and also applied this to the preparation of the galicaftor cyclopropyl fragment in 95% yield in a single step. Moving on to C2 correctors, I mentioned the prolidine series as one that we were particularly interested in. In this case, a high-throughput screening campaign identified this hit, and unfortunately, while the activity was quite good, the properties of this aryl ketone were quite poor, as you can imagine. So there was a desire to expand this into ether and amine-linked compounds. And so, as embedded members of this team, we thought that nitroolefin 3+2 could be a good approach to access both amines through just simple reduction to an amine, or through some kind of Neff reaction to give alcohols and then ultimately ethers. So with this as a strategy in mind, we scanned the literature and found very few examples of alkyl nitroolefins successfully engaging in asymmetric 3+2 type transformations. This paper in particular had just one example of an alkyl nitroolefin, which was isopropyl. All others were aryl, and that was really common across the literature for this kind of reaction. Fortunately, we found that conditions that were quite similar to what was reported here, although we switched from perchlorate to triflate counterion, cut down the catalyst loading by a factor of 20, and found that utilization of catalytic amount of base was highly effective at modulating this reaction, gave excellent EE and reasonable endoaxal selectivity with endoisomers that could typically be separated very, very easily from the exo. So this proved to be a remarkably general reaction. In this case, many bulky nitrile alkene substituents, as well as many aryl imines, were successfully utilized as substrates in this chemistry. And a total of over 1,600 analogs were prepared, where we would typically make quantities of a core that would have 4 stereocenters, and then our high throughput chemistry colleagues would make libraries of compounds from that, and really exploded the SAR opportunities here. And something that was really remarkable to me is substituents that you may consider potentially offensive to copper catalyzed reactions, like this dimethylaminopyridine, were actually the highest yielding and most selective examples. So this was a really good general reaction that impacted this project. So as the project progressed, there was a need to make gram quantities of lead molecules that had promising data. So these are just examples of some of the structural diversity of molecules that we prepared between a 10 to 50 gram scale to support advanced preclinical characterization. And something else that we've had an opportunity to do a number of times is have members of the group visit process chemistry as members and serve as members of the API team for some period of time, a few months. And Steve Gressler from my group, who's a co-author on the innovations article, spent a little time in process chemistry helping to scale the first third of a kilo of ABBV3221 after it was selected as a candidate for further development. And really that collaboration with Process Chemistry Discovery Support Group is really very, very key. And they helped identify some new NEF reaction conditions here to convert this nitropyrrolidine to ketone. We were using excess stoichiometric chromium dichloride, which was obviously not very good for scale-up. And they identified a really nice DBU carbon disulfide adduct that replaced that and really helped us to scale this compound in a more environmentally benign way. So I'd like to step back for a moment in the middle of the C2 corrector story. And since, I've really enjoyed Dani and LC's opinion in Nature Chemistry on the importance of academic collaborations. Really helped to validate, I think, something that we've believed in for a long time, engagement with the external scientific community. One way that we've engaged with this community is through the NSF Center for Selective CH Functionalization. This is a collection of more than 20 faculty at over 15 universities across the US, really focusing on paradigm-shifting research in CH functionalization. What we really like about this group of researchers is their focus on collaboration instead of competition, which used to be somewhat rare in organic chemistry. They embrace industry engagement, which is also highly valuable to solve real-world problems, not just esoteric academic problems, but really trying to apply and find solutions that will be useful to practitioners of organic chemistry like us. It's given AbbVie some access to top talent and certainly early view into cutting-edge research. And they also champion a number of really important initiatives, such as the Network for Diversity in Chemical Research, which provides some long-term mentoring for faculty at schools that may not normally have opportunities to be engaged in this kind of collaborative multi-institutional research center. So we started collaborating with the CCHF around 2015, and for the first few years, we're engaged in a number of pre-competitive collaborative projects with Hugh Davies, who's the director of the CCHF. Since that time, we've expanded to collaborate with 5 different members. All these asterisks are the different group members, including Jin Kwan Yu, Shannon Stahl, Donna Blackman, others who are part of the CCHF. So really, we've benefited in many ways from the CCHF collaboration, opportunities to present externally, collaborative publications with the Davies group, hired a total of 12 people from 8 different research groups. We've had a lot of nice, unique research learning experiences with students visiting AbbVie, scientists from my group visiting Emory. Also, Hugh sharing a short course on the topic of CH functionalization a few years ago. And really, with the research, the pre-competitive research, we were able to selectively functionalize a number of CH bonds that previously could not be selectively functionalized. And this really helped to create a number of very interesting building blocks and opened up chemical space that was previously inaccessible. So to remind you of, again, this article, I just wanted to snapshot this. Specifically, Dani and LC highlighted the gap between, I mentioned the gap between discovery and process. There's another gap that Dani and LC talked about in this article between fundamental academic research and the applied industrial research that's necessary to ensure real-world application. So there was a call here to engage academic scientists and have this honest and maybe uncomfortable discussion about, will my chemistry ever be adopted? This is another cartoon, actually, that was from this paper, where the academic in the ivory tower is saying, why don't you use my catalyst? And the chemist is saying that the ligand takes 15 steps to make, so we're never going to take a week or 2 week research project to make this ligand for a reaction that may or may not have any utility for our chemistry. So how is this relevant to the Davies collaboration? So after having these pre-competitive collaborative projects over several years, in 2019, an opportunity came about where we had some chiral cyclopropanes on this C2 corrector project that really the existing chemistry that Hugh had published over the last 30 years in this area had some gaps that we recognized. So there were many heterocycles that were not tolerated in this chemistry, often due to the fact that the catalysts were only effective in hydrocarbon solvents. So in order to advance lead compounds, we really needed an asymmetric synthesis. We started with a method that utilized a pentolactone auxiliary. While this was effective, it was steppy and not very atom economical for scale up. So there was a recently published method from Hugh's group that used very low catalyst loadings to promote these reactions. And this was actually surprisingly or very effective across many heterocycles that hadn't previously been demonstrated in this asymmetric cyclopropionation chemistry. However, there was one notable exception, which was ortho-substituted aryl diazoacetates, which in this case, with this one that we really needed, this orthomethoxy example, was very poor yield and poor in antioselectivity. So since the Davies group had an arsenal of many catalysts available that had been published for all sorts of different purposes, and we already had this collaborative relationship established with the group, we asked if they would be available to take a look at this problem. And they found very rapidly, in a way that impacted the medicinal chemistry project in real time, new reaction conditions that, in this case, took this specific molecule from a 30% yield to an 87% yield with almost perfect and anti-selectivity. We published this story earlier this year. So what is so unique about this catalyst system? So while this catalyst had been published for a number of CH functionalization reactions, there weren't any examples that we were aware of where this had been used for cyclopropionation. And very interestingly, let me reverse for a second. We found a critical factor where the 2-chloropyridines worked incredibly well, but other heterocycles that did not have this substituent did not give high selectivity. But it turned out that just including 2-chloropyridine as an additive actually solved this problem and gave universally high selectivity in these types of reactions when there was an ortho substituent. So it turns out that if you take the catalyst, you can actually grow a crystal structure of the catalyst with the 2-chloropyridine bound. And we found that this perturbs the structure of the catalyst and really allows optimal interaction with these ortho-substituted compounds. And just to bring you back to this cartoon, the thing that we really liked, one of the things we really liked about this is the ligand takes a single step to make. So you just take L-tert leucine, abundantly available, heat it up with tetraphenyl phthalic anhydride, and that's one step to the ligand, not 15. And just do a ligand exchange with rhodium acetate, run a column, get a nice blue band of your rhodium catalyst, and you can get it in about an afternoon. So it's really, really a nice example of a practical catalyst. So without the collaboration, we probably wouldn't have found this, since it hadn't ever been published for this use before. And really all these additive effects and things took a lot of innovation from the Davies group in partnership with AbbVie chemists to figure this out. So circling back to the C2 corrector project, what does this have to do with cystic fibrosis. So one of the C2 corrector series was this cyclopropane with the orthomethoxy substituent. And we found that substituting the cyclopropane gave greatly enhanced activity. And the chirality had a very significant effect on the activity, one enantiomer being much more active than the other. So since chirochromatography was required, it was very slow getting through the SAR here. So the method that I just described to you greatly assisted SAR exploration and allowed preparation of material in sufficient quantities to push through the screening funnel. And all sorts of molecules were accessible with this method. And not only that, when the CFTR C2 lead molecule emerged, our process colleagues were able to, with not a lot of changes, implement this chemistry, in this case using 2-fluoropyridine instead of chloro, in order to prepare 2.5 kilos of the chiral cyclopropane intermediate using flow chemistry to address the expected diazo safety concerns that were very effectively addressed through flow chemistry. So this was a real-time impact on both the discovery and process chemistry project with an academic collaboration. So moving on to much more brief case studies in both Parkinson's disease and hepatitis C. I wanted to describe to you a really interesting project that was very unlike the one that I just described to you. It was a series of more traditional SAR development campaigns to find a lead molecule. So this was a project where we wanted to address unmet needs in Parkinson's disease. So Parkinson's is caused by dopamine deficiency in the brain. And this schematic here describes a somewhat complicated biochemical pathway that gets dopamine into the brain. So levodopa in the periphery can cross the blood-brain barrier. However, in order to turn levodopa into dopamine, you need to decarboxylate it. So levodopa itself is a prodrug of dopamine. However, you don't really want this to happen in the periphery. You only want it to happen in the CNS. So if you're trying to replace deficient dopamine via treatment with levodopa, it's important to dose a dopamine decarboxylase inhibitor, such as carbidopa, to prevent peripheral decarboxylation so that as much as possible, L-dopa can enter the brain where you want it to be decarboxylated to form dopamine. So with this, another issue, besides the complex pathway here, is that levodopa and carbidopa both have very short half-lives. So with oral dosing, which has been used effectively for about half a century, of course, you have highly fluctuating drug levels. And the problem with that is that high levels of levodopa can cause dyskinesias, undesirable side effects, especially as the disease progresses to advanced Parkinson's. And low levels, of course, also have problems because you don't have enough dopamine. So you get this early wearing off or delayed on time period phenomenon. So because of this, it's very beneficial to sustain a constant level of levodopa. And for that purpose, AbbVie put forward Duopa, which is approved for infusion, continual infusion. However, this requires a surgery for daily intestinal dosing of 100 milliliters a day of gel because of the insolubility of these drug substances. So one approach to addressing this unmet need would be to have a more highly soluble form of levodopa and carbidopa. So one idea for this was to make a phosphate prodrug. And while there were some examples of phosphate prodrugs of phenols in literature, there's still gaps here that hadn't yet been filled in considering what impact that could have on Parkinson's disease. So there was really an opportunity for synthesis innovation here because of the stability and isolation challenges in making these very highly polar, highly functionalized molecules. So initial synthesis and assessment of these identified levodopa 4' phosphate, carbidopa 4' phosphate as optimal. and these were named phospholipidopa phoscarbidopa. So the first-time synthesis of levodopa phosphate is shown here. There was a non-selective benzylation that required a very tedious separation, and then a final phosphorylation and debenzylation. So while this was useful for preparing some quantities of the levodopa phosphate, it failed to provide monophosphates of carbidopa. So we identified an improved Negishi coupling route that provided the levodopa phosphate very efficiently in a regioselective manner. So this was effectively used to increase the yield from less than 3% to over 50% with this Negishi coupling methodology. So for carbidopa, the way that we first made this was the single-step direct phosphorylation, which gave low yield of a mixture of 3' and 4' phosphates. We could separate these by HPLC separation. However, this was not deemed suitable for larger scale synthesis. So we later did find more scalable ways to make these and did collaborate with process chemistry on that, and they published that story. A big step forward, however, with carbidopa phosphate was the isolation of, when we first made this, concentrated the aqueous HPLC fractions. We got a really nice solid, and this turned out to be a crystalline, stable crystalline trihydrate form that in very high purity that was quite stable. And so this was a big step forward for considering this as a development candidate, and to encourage us to consider the possibility of large-scale API production. And there's a lot more details in this recent JOC paper that was just published. So I guess I did want to mention that we're very excited that just last week actually pivotal phase 3 results were published for phospholipidopa phoscarbidopa. So we're really looking forward to seeing how that therapy can help patients. So my final case study before I conclude is in the area of hepatitis C. Rather than going into the disease in detail, I just would like to refer you to our Mavorite homepage, where you can see highlights of Mavorite, which is the only 8-week cure for all types of hepatitis C across genotype. It's currently the #1 prescribed chronic hepatitis C medicine. It's really a remarkable advance for patients suffering from this disease. It's a major global health concern that primarily affects the liver, eventually leading to cirrhosis and cancer. So being able to cure this disease has been really a breakthrough in modern medicine. So from a synthetic chemist point of view, many antiviral medicines are in this beyond rule of 5 space. So well, I don't expect you to be able to see these structures here. All of the purple highlighted structures, so we've partnered with Jan Mjarderson at the University of Arizona to use many of these top pharmaceutical posters that we often use as artwork around our labs at AbbVie. And this one in particular, we thought it would be interesting to publish all of the beyond rule of 5 orally available drugs that have ever been discovered and created. And out of all of these, all the ones in purple are antivirals. So there are many of the beyond rule of 5 orally available drugs are actually antivirals. And 5 of those are shown here. 3 in Vicurapac, our first approved HCV medication, and 2 in Mavorite. And out of these, you'll notice that Fibrentosphere is particularly large and lipophilic, with a molecular weight over 1,100 and C-log P of almost 10. So this is in far beyond rule of 5 space, really the world record holder in de novo designed molecule size. But because of its exquisite potency, it's very effective as a component of Mavorite. So despite that fact, we thought that if we could take another look at a solubilizing prodrug approach, of course this is very different. This is not a phenol. There's no phenol or any alcohol at all to make a soluble phosphate. So it was really a challenging problem to consider how would we enhance the solubility of Fibrentosphere. So a heroic campaign to prepare many different prodrugs identified that alkylated benzimidazoles could be prepared and isolated as stable materials that contained a phosphate in a group that would come off once phosphate cleavage took place. So members of the group were embedded in this discovery team and really enabled routes to several classes of fibrinosphere prodrugs. And this was especially challenging because of the C2 symmetry of this. So these benzimidazoles, there's an up and down nitrogen, and both of these are homotopic equivalent. So selectively functionalizing this was really tough. And our first time synthesis of this of one of these molecules is shown here, where we did a non-selective alkylation and very low yield. And then, taking off benzyl groups gave the final prodrug. Unfortunately, this was only 11% yield from fibrinosphere, which is a very valuable molecule. So when we brought forward this molecule as a potential development candidate, we were challenged to find a route that was closer to 50% overall yield before it really could be considered seriously as a development candidate. So this was seen as a very significant challenge considering the structural complexity of this molecule. So really the problem arises from the similarity of these nitrogens. So to tackle this, we thought if we could just protect one of these benzimidazoles selectively somehow, even though these are identical homotopic benzimidazoles, and then carried out a regioselective alkylation and deprotection, we could access all of these molecules using from a key intermediate. Unfortunately, our first attempts to protect this, not surprisingly, gave exactly what you'd expect, a statistical 1 to 2 to 1 ratio of starting material to mono to di, with every conceivable up and down isomer that you would expect from this reaction. So after tedious separation of the mono-Bach isomers, we had a serendipitous discovery that the down-Bach isomer was much less soluble than the up-Bach, which would prove advantageous in efforts to find a better synthesis of these molecules. So in order to solve this problem, we incorporated an unusual application of the Hero principle of statistical amplification. So by simply cooling down the Bach protection, we could get a 5 to 1 ratio of down to up Bach, which still wasn't great. But by letting this reaction go to complete di-Bach, each of these molecules went on to give a 5 to 1 ratio of down to up Bach, giving in the end a total of 97 to 3 ratio of substances containing a down Bach versus those that did not contain a down Bach, which is only this up-up di-Bach isomer. So this would only be relevant if we could selectively cleave the up-Bach isomers. So in the same pot, we could just switch solvents and introduce butylamine. And you can see here this intermediate 69 to 28 to 3 ratio of these 3 di-Bach species. And by introducing butylamine and allowing the down mono-Bach to precipitate, we could drive up the formation of this isomer and ultimately get, with slight change in conditions with another solvent switch to optimize that the D-Bach rate difference phenomenon in acetonite trial, we could actually get 88% or 85% isolated yield of the down mono-Bach, or up to 94% with a resubjection of mother liquors. So with this, we showed unambiguously that we had the right isomer through an X-ray crystal structure, and then we're able to advance this key intermediate after hydroxymethyl formation, a second crystallization-induced selective benzimidazole alkylation with paraformaldehyde. This key intermediate could be advanced to all 3 prodrug classes. And in the interest of time, I'm not going to go over any of the details here, but we did publish this paper earlier this year if you would like more information. So this was really SAR enabling and greatly improved the yields of all the lead candidates in this project, and therefore really met the requirements that development has set out. So to conclude, we put forward that we and others across the industry have identified a new role for organic chemists beyond just discovery and process, which is the role of a discovery synthesis group. And one of AbbVie's discovery synthesis groups, which is COS, in its first 10 years, it's contributed to the advancement of over 30 candidate molecules across AbbVie's portfolio. What I showed you today was by no means comprehensive. These are just publishable stories that we were able to publish. So all of these projects actually were completed from the start of the project to finding a candidate in less than 2 years. So it's generally thought that the industry average is around 4 to 5 years. in discovery. So by no means do we claim to be the only cause of that acceleration, but we do believe that having embedded discovery or having embedded synthesis specialists in the teams did help to accelerate these projects. And as I said before, the CF project really best illustrates the most prototypical mode of COS chemist embedded collaborative engagement as part of a medchem team. Although we've had opportunities for other unique projects, impacting projects in other unique ways. So with that, I'd just like to thank everyone who has been a part of these projects. This is an outdated pre-COVID picture of the group from a couple summers ago. This group's changed a lot since this time, but just like to thank all past and present COS group members. Just an amazing team of chemists. It's really been a pleasure to work with over the years. And specifically, I'd like to call out Steve Gressler and Phil Kim, the head of CMEC, who helped me write this innovations article, and really have been involved with the group from the start, and been this very important part of COS over the years. I'd like to thank a list of literally many hundreds of AbbVie scientists across many projects that I highlighted today, and also our academic collaborators, specifically Jan Yarderson and Professor Hugh Davies at Emory University. With that, thank you for your attention, and I would be happy to take any questions.
Thank you, Eric. Really great job. And it's a good thing that you acknowledge all of the hundreds of chemists at AbbVie, because I think most of them are on the line. So good cover there. While people put in their questions, I'm going to maybe kick things off. And this is geared towards your collaboration with Hugh. And I think it's a really great story. And I think it's interesting because it seems to be a collaboration that occurred along the critical path of drug discovery. And I guess I always imagine that to be a fairly complicated way to navigate. And so I guess what was successful for you in this collaboration with the Hugh Davies lab?
So I guess you mean with respect to kind of the logistics of getting things arranged?
Yeah.
So I guess that's where the value of the pre-competitive relationship really came in, because we already had, collaboration agreements and all of that in place. We had an ongoing collaborative relationship with the group. So when this opportunity arose, it was really a seamless thing to just say, we have another project, let's pivot and do something that can really impact the project. So that was it's not CH functionalization, right? But it was something that they had a lot of expertise in that we couldn't seem to figure out a solution for. So because we already had this established relationship, we could just say, you guys try this out and see what you can find, and maybe we can impact the project. And it's just remarkable how it all unfolded.
Yeah, that's great. So a question from the audience is about, I guess, the utilization of enabling technologies such as like photochemistry, electrochemistry, flow. You kind of highlighted a few of these throughout. I guess, how do these come into play within the discovery synthesis groups with respect to speed, but also, with line of sight towards a greener process?
Yeah, that's a great question. So we have very close internal collaborative relationship with our Advanced Chemistry Technologies group led by Ying Wang. So that group has just incredible expertise in applying new emerging technologies to impact discovery projects. And so we very often will engage with their group in order to explore any sort of technology-enabled synthesis. And we will then definitely take things on and incorporate it in our own labs. But very often that's a way to sort of get a kickstart anything that requires some special technology. And they have, so parallel high-throughput experimentation, capabilities, technology, expertise that really can help to identify new reaction conditions rapidly, whether or not it requires technology.
I guess, are there any technologies that you would like to see brought into the DSG?
Yeah, I think we still need to do, we could do a lot more with electrochemistry than we do. I think there's, been a continually exploding opportunities there that I don't think, I think the industry is yet to fully embrace some of the opportunities there. although there are certainly challenges associated with that. Definitely I think the reaction miniaturization concept, there's a lot of opportunity there, and I think just limitless opportunities for simultaneously assessing thousands of reaction conditions simultaneously to find an answer to a key synthesis problem question very rapidly. That I think we still have room to grow there. And also, I think the topic of the first webinar, artificial intelligence, machine learning. I think there's tremendous untapped potential in our ability to predict reaction conditions or synthetic pathways without so much brute force, empirical sort of step at a time, figure out how to do it. So, so far it doesn't seem like there's a robochemist that can come anywhere close to what we're doing with the way that we're doing things now. Although there have been tremendous advances in this field, and I think we really need to stay on top of that as well. As an important area that will certainly greatly influence how we make molecules in the future.
That's great. Okay, slight pivot. With so many projects, at what stage does the DSG engage with the medicinal chemistry groups?
So we are embedded very early on. So our most, I would say the place where we can impact things the most significantly tends to be in the more late discovery space, where things are getting closer to finding a candidate, because the synthesis matters more and more, right? The later a discovery project is, the later phase that discovery project is in. So that's where I think our engagement with the team can be the most beneficial. However, we are often engaged, before a project transitions to lead op, really trying to find, as I like to call it, a synthesis-driven design, where sometimes synthetic accessibility can create ideas that maybe an in-depth understanding of the receptor biology or SAR wouldn't. So there can be influences in direction that may not have been predicted through other kind of modeling tools or other techniques, that if you can open up accessibility molecules that look like, resemble those that seem to be active for a project, you can end up in interesting chemical space in a way that's more convenient to prepare, or more gives you more novel chemical matter. So however, we've been engaging earlier and earlier. Actually, we have been very involved in the DNA encoded library space over the last couple of years, engaging in even pre-target identification, synthesis advances, right? So in the very earliest stages of drug discovery. So, and of course, I showed an example of being part of process chemistry teams as well. So I think it's a really exciting place to be, because we really can span the gap, or can span engagement from the earliest stages of discovery all the way into development sciences.
So to kind of follow up with that, it seems like DSGs are really great at the bigger companies, AbbVie, Merck, and I'm sure others as well. And a question came in about how do you think about scaling these to, say, a more smaller company with less resources available?
Yeah, that's a good question. I mean, I would argue that any company, no matter what size, would benefit from having at least a couple of synthesis specialist chemists who are told that you don't need to be the SAR expert. We just want you to focus on solving synthesis problems, and but not be a process chemist. We don't want you to run this. We don't need you to focus on the perfect way to run this process. We just want you to address whatever bottlenecks there are that are slowing the discovery team down. And I would say that any company, whether you have 5 medicinal chemists or 500 medicinal chemists, I would argue that anywhere from 10 to 30% of that those chemists, if they were deployed to focus on synthesis, that could be a real benefit to that organization.
So to kind of follow up on that, and this might be the last question, is, I guess, what place do you feel that these discovery synthesis groups also have amongst like the biologics and also the vaccines space? Like, can it be expanded to those arenas as well?
Yeah, it's a good question. There, I think so. An area that we did focus on is in the context of antibody drug conjugates. So there's a great need for innovation there that was really a big synthesis problem, and I didn't have any stories to share from that effort. There's certainly opportunities across different modalities that where synthesis innovation is very beneficial. Everything that I focused on today was really in the small molecule space, but there are definitely opportunities outside of that, and we've been engaged there as well. So, I think anytime that accessibility to substances is a rate limiting step, as you nicely introduced, I think a discovery synthesis group can certainly be beneficial in providing more rapid access to that chemical matter to accelerate a project's progression.
Thank you, Eric.
Yes, thank you very much, Eric and Dani. A great presentation today. And I would also like to thank you, the audience, for attending today. I hope this was insightful. And again, thanks for joining. Have a great day.
Bye, folks.